asexual reproduction, we'll be looking at fungi and protists. We'll also be looking a little bit at some sexual reproduction in fungi to understand the difference between when they'll choose to do asexual and sexual, and also just we'll be putting a few other things together with that as well. So just the outcomes that you can pause and look at. Okay, so fungi. So fungi are eukaryotes. Now they have cell walls, and single-celled fungi, when they're aged, can have large vacuoles, which can make them like plants, but they are actually more like animals. So they're heterotrophs. They need to obtain their nutrients from their environment. They don't make their own energy, so they have no chlorophyll. They don't do photosynthesis. They often act as decomposers in ecosystems. So looking at asexual reproduction in fungi. So fungi can range from microscopic, single-celled, like yeast on the bottom left. They could also be mold, which is like the furry stuff you'll see growing on bread or fruit when it gets old, and mushrooms. So we'll look at budding in yeast first. So yeast are a unicellular fungi. So one common example is Saccharomyces cerevisiae. It's commonly referred to as baker's yeast. So it's used in research as a very simple model eukaryote, also used in food, in terms of like baking bread, also used in making beer and wine. Now, most yeasts, including Saccharomyces cerevisiae, reproduce asexually by budding. So we'll have a bit of a closer look at budding. So we'll just be starting off with a single cell. The red denotes the nucleus. Now budding just starts by basically a little lump will start forming on the surface, and it will grow. Nucleus will migrate to the division between the newly formed daughter cell, or the newly forming daughter cell, and the parent cell. Then we'll just have mitosis, where the nucleus will divide, and eventually when the daughter cell has grown big enough, the two will separate, and we'll have a daughter cell and a parent cell. The daughter cell will continue to grow more until it reaches a size big enough where it then may become the parent cell, and a new daughter cell will bud off there. So this just shows you what this would look like under a microscope, just looking at yeast again. So firstly, not every cell may look like it's budding. So we see these kind of three there are just kind of on their own, but we see these two here at the bottom left. We have a bud starting to form. It's a bit bigger there. This again just kind of shows you many examples where we'd have a daughter cell kind of budding off an adult cell. Again, not everyone. I mean, the other thing that kind of muddies the water, yeast can be a bit sticky. They'll stick together as well. So if we see two big cells together, one may not necessarily be a daughter cell, but let's see, the last thing is when they do bud and the cells separate, there's a thing called a bud scar that forms as well. So this is just an example where they've used either a fluorescent stain or a fluorescent protein just to specifically show up where the bud scar would be. So just looking a bit more at fungi. So except for yeasts, which we just looked at, which grow as single cells, most fungi grow as thread-like filaments called hyphae. So the hyphae consist of one or more cells surrounded by a tubular cell wall. A mass of hyphae coming together makes up the body of the fungus, which is called a mycelium. Now, an example of a mycelium is the fuzzy mold that grows on bread and fruit. So this just kind of, I guess, starting at the bottom right, this shows you what we were kind of talking about. So we've got the individual hyphae, which would be these tube-like structures. But when all the hyphae comes together, it forms the mycelium. And what we see as mushrooms, just note, is just the above ground reproductive parts of the fungi, where most of it is, most of the action's happening underground. Just moving on to this one, we've seen yeast, the single-celled, where something would be budding, and mold, we see the hyphae there, where again we just have, it could be one or more cells with a tubular cell wall around it. So looking at reproduction in multicellular fungi after looking at single cells. So remember, single celled fungi were reproducing by budding. Now we'll look at multicellular. So most multicellular reproduce by forming spores. So the mushroom seen in multicellular fungi, as I said, is the reproductive structure that releases the spores. And we just kind of show a couple of pictures here where, we've got the mushroom and put it on paper. You see the spores kind of have formed down there. This is a particular type of mushroom that will just shoot its spores up into the air for it to be dispersed. So a bit more about what are fungal spores. So we can think of spores as being a little bit like seeds kind of for the fungi. They're not exactly seeds, they are different, but they do have a few things in common with seeds. So they're small and they're light and they can be dispersed by wind, water, and animals. They can also last a long time as well. They don't have to be in water. They don't have to worry about drying out. They can kind of just sit around for a while until conditions are favorable where they can then grow. They'll be produced in large numbers, which is a strategy often needed if it's going to be dispersed by wind or water or animals. So they can kind of be dispersed somewhere else and then can germinate in a new location where they can grow into a new fungus. And now just noting that spores can be produced asexually, sexually, or both. And just having a little look at some of the structures involved with spores and making spores. So we have the sporangium here, noting that it's kind of like on a long stem, and I guess similar with plants. If you want something dispersed, and it will be better, or you've got more chance of it being dispersed further by the wind if you can kind of elevate it a little bit to get more exposure to wind. So we'll have a sporangium, it will burst, kind of releasing all of the spores into the air, which will hopefully then get blown away for it to grow somewhere else. So the formation of spores. So multicellular fungi can produce haploid spores. Remember, haploid meaning that there is just one copy of the DNA, haploid half, and it will produce these spores by mitosis. So this is asexual reproduction, with the spores containing an exact copy of the DNA from the mother organism. So remember, with this, there is no deliberate creation of variation. If it's asexual, if it's mitosis, we're not deliberately trying to create variation. So what about sexual reproduction in fungi? So two fungi can temporarily fuse to create a diploid structure. So this structure can then produce spores via meiosis. And so we'll pause there and take a step back and say, so why can't the haploid structure produce spores via meiosis? We think, well, it has to be diploid. So in deciding which it's going to be, we'll just think if it's haploid, then it can only do it via mitosis. If it's diploid, then we can do meiosis. So meiosis creates variation by having DNA from two parents, plus there's additional variation that comes from independent assortment and from crossing over, which you'll see when we deal with meiosis in detail. So this just kind of shows you a cycle of where we may have a diploid fungus kind of producing spores via meiosis. So we'll start at the bottom here. Now, importantly, there are two mating types, that here is just denoted with a minus and a plus. We can kind of think of this, it's like being two genders. It's not exactly that. It could just be as simple as just the two different mating types will be expressing one of two different proteins, but there needs to be two different mating types, just kind of like it's a male and female. So they can come together to be forming a diploid structure. And then through meiosis, they can form spores. The spores will be released from the mushroom. So now again, we have all of our spores that have been released. We've got the two different mating types, the negative and the positive. And again, they can then come together again and the cycle will continue. Okay, so what we did so far is the basic stuff to know. I want to take you through just a little case study involving Saccharomyces cerevisiae that kind of puts a few of these things together. But before getting into this, I just kind of want to point out that this is kind of like a little bit extra. Don't think that this information, I guess, is equally as important as everything that I've dealt with so far in this video. If you have to prioritize, just nail all of the fungi stuff up until here. If you need to kind of write notes and memorize more about this Saccharomyces cerevisiae case study, just make that the icing on top of the cake. But just focus on everything that I've done up until this, and this is a bonus. Okay, so Saccharomyces cerevisiae, it's a single-celled yeast. Now, it can reproduce using asexual reproduction via budding, but it can also reproduce sexually with spores formed via meiosis. So I guess one of the obvious questions that then comes from that is, how does it choose between each strategy? Is it random, or is there some sense or driving force to it if it can use any one of two methods? So firstly, let's just say have a few basics that will help us understand how we'll answer this question. So yeast can be haploid or diploid. So haploid yeast cells will grow by budding. And when two haploid yeast cells of the opposite mating type meet, they can mate and form a diploid cell. So I'll stop my sharing and just kind of draw a bit of a picture to show you. So if we start off, we'll have two haploid yeast. So I'll say haploid. And also just denote the haploid just by having a single N. Often the two in front of the N will denote diploid, and just a single N will denote haploid. And I'll just say they're the two mating types, which with Saccharomyces cerevisiae is just denoted by having a mat A and a mat alpha. Now, so they on their own can just can reproduce asexually via budding. So they can just kind of keep budding and everything will be fine. But if we have the two opposite mating types come together, they can come together to form a diploid structure. Or a diploid structure. They can form a diploid yeast. And again, if conditions are good, it can still keep reproducing by budding as well. And in fact, in a large population where you've got them growing, you may have a mix of all of these happening. Pardon me. If we've got, for example, just some mat A haploid, if they don't encounter the opposite mating type, they will just stay haploid and they'll just keep budding. Same with that. Whereas if they have two come together, then we'll have diploid, and it will just reproduce via budding. So that can continue like that for a long time in good conditions. But what happens if conditions change? If we start, we start using up all of our media, it starts running out of food, and we get into starvation. Well, it turns out that starvation is then the trigger for it to make spores. To make spores via meiosis. So it gives you an elegant explanation as to why it may use budding and why it may use meiosis. So if we forget about the starvation, so let's think conditions are good. And if we think back to asexual reproduction, one of the advantages of asexual reproduction is simply that it does not have to expend extra energy or effort on sexual reproduction. If conditions are good, there is no need for genetic variation because it can cope with everything and everything's going fine. So it can just very quickly reproduce asexually, build up big numbers very quickly to take advantage of that. But one of the drawbacks, of course, of asexual reproduction is if conditions turn bad and there is some kind of selection pressure, then they don't have the genetic variation that may increase the chances of a variant surviving that selection pressure, which is where they then have to think of a different strategy. So now we have starvation. Now we have a selection pressure that can threaten the whole population. So they can change their strategy. They can now make spores via meiosis, where they can introduce variation. And by introducing that variation, they now have variation in the population that increases their chance that a variant will now be able to survive the selection pressure of starvation. So we'll go back to what we were looking at. Yeah, so which is what I've got here. So in good conditions, the diploid cells will grow by budding. In starvation conditions, the diploid cells will form 4 haploid spores via meiosis. So this is just another way to show that. So we've got our mat alpha mating type. It's just reproducing by budding. Everything's fine. Our mat A, it's just reproducing by budding. Everything's fine. If they come together, we don't need to worry about the conjugation part. But if they come together, they can form a diploid. But again, if conditions are good, the diploid can just keep reproducing via budding. Everything's fine. But then if we get to starvation conditions, at this point, instead of going around and reproducing asexually by budding, it has then decided to use a different strategy that has been triggered by starvation, where it will undergo meiosis. It will form 4 different haploid spores, create variation, and then these spores will then kind of form new haploid cells that can mate, and then hopefully survive the selection pressure or survive the starvation. Or simply because they've formed spores. Spores are hardy structures that can then just sit there and wait out the tough times, and then if good conditions come again, then they'll be ready again to grow, and the species can live on. So just highlighting there that it takes advantage of the best of both worlds when it comes to asexual and sexual reproduction. So they can take advantages of asexual reproduction by just moving really quickly and reproducing in big numbers very quickly. But they can also take advantage of what sexual reproduction offers in terms of having a large amount of variation or increasing variation, which increases their chances of surviving some kind of selection pressure and undergoing evolution. Okay, so now let's get back on track. Now we need to look at protists. So there's only a few slides on this. It's not a lot of information, but the syllabus outcomes do require us to know the protists, to know an example of them. So protists are unicellular eukaryotes that do not fit in the animal, plant, or fungi kingdoms. So they're very diverse. They have a range of structural forms, modes of nutrition, habitat, and reproduction. Look, another way we could interpret that point is that it's very complicated. It's kind of beyond the scope of this course to really know a lot of detail about them, just because there's just too much to know. Now, some examples of protists include amoeba, paramecium, and giardia. Some protists use binary fission to reproduce. Just remember that is similar to reproduction in bacteria, in prokaryotes, and some use budding, like what we have seen already. Okay, so some protists such as amoeba and paramecium use binary fission to reproduce. So as I said, just like bacteria. So if for example, you were to have a question, give an example of a protist that reproduces via binary fission, you would need, or name an example. Well, there's a couple here we could use. So we could say amoeba and paramecium, and binary fission, it's just we need to divide the DNA in the nucleus first. Once we've got 2, we're just going to split the cell right in the middle to get 2 cells. Similar thing here. And some protists reproduce using budding, like the single-celled fungi. So in budding, we're just remembering a small outgrowth develops from the parent body, which separates and develops into a new individual. And again, the named example that we would need to know and remember we have here is Arcella, which is a sarcodine. So the budding's different. The budding is the same as what we've seen. We're just using it with a different example of the organism. So yeah, protists, Arcella. And I think that's it. There we go. Yes, that's it. Thank you.